Then what you have to know before starting the chapter, so that is how the gametes are formed. You also have a brief idea of how the gametes are formed. So that is through the process called as gametogenesis. You already learned the mitosis as well as where the meiosis is the gametogenesis. Right. So what happens? So you can take an example of a human body. The cells within human body is composed of 46 cells. Right. So these 46 chromosomes, whether it may be a what is? So how the gametes are formed? Here there are 46 chromosomes in the human cell. So where they undergo a process called as gametogenesis. Right. So what is this gametogenesis then? So gametogenesis as the name itself has gameto, that means the gamete, genesis, that is the formation. It is the process of formation of gamete from the diploid reproductive cell to through the process of meiosis. So that is what we call as gametogenesis. So in the human cell there are 46 chromosomes. The gametogenesis chromosome, then 23 chromosomes will gather, then it can zygote, as the zygote undergoes process where the number of cells are multiplied and body as the first cell, there are 12 cells. The gametogenesis sperms are formed. Then egg cells are formed. The two together they will form what? A diploid zygote. This diploid zygote can be so other definitions you have to know before starting the chapter is about allele as well as allele. Then what are the alleles called as? So you already know genes. The genes are the vehicles of the genes have all the genes have all the information that makes an organism character. So what is allele then? So allele is nothing but is a contrasting form of a gene, also called as what? Terminating form. So what is that allele means? So you can take an example where the gene which goes for a height, so that is what height that is called as a height gene. So where that height gene has different forms of those genes. Height has a different forms of alleles. So what are those alleles? So height may be tall or height may be dwarf. So that means the allele may be tall or allele may be dwarf. So that are what the alleles of what the gene coding for height. So you can see so there are different types of alleles. One is homozygous allele, another one is heterozygous allele. When it comes to the homozygous allele, so there you can see where since the genes can't be seen with the bare eye, so they can be represented with this capital what tall allele. Right. So again, multi that indicates the alleles are one. So when it comes to the heterozygous allele, one is capital T, another one is small t. It also codes for the tall character of an organism. So what are the differences between character traits? What is this? And codes for a particular character, so that is height. And this height is has the different alleles. What are those? One is capital T, another one is small t. So that represents a different height, such as tall as well as short. So those are called as what? The alleles. Right. So now we will know the differences between the character. Tell me what are the differences? How you can differentiate character from a trait? So when it comes to the character, these are the features of the organism which have which are of a different character. You can take an example of one feature or a character, eye color. When it comes to the eye color, so that is a character, and the expression of those characters, the expression of those characters in the organism, that are what we call as characters to the substance of the organism. When it comes to the traits, the expression of those particular characters in the organism, how it is expressed. Eye color is a character, that how the eye color is expressed in case of organism, so that is what we call as. So you can take an example of trait. The eye may be black in color, eye may be blue, eye may be brown. Sometimes you get green eyes also. So that is what we call as trait. Character, the feature of an organism is eye color. So the expression of those character of eye color, so that is what we call as trait. The trait may be right. So that is black eye, blue eye, and brown eye. So next we can take an example of height. Height is a character which goes for the traits which have different traits such as tall as well as short, intensity, different height. So that is what we call it as traits of character height. So if you take another example of character, that is hair color. Hair color also varies. It also has different traits. One is that these are also the traits of character. That is what the right. So there, these things character of their height. The next one is the expression of those features in the organism that are height, that tall or short. Eye color is that black, brown, brown, that is contrast form of alternative form of when it comes to character. Those of the organism. So that is the tree. The express those characters is called as what? Trees. This is what so that is next you have to know is about that is a dominant trait as well as specific trait. The name itself is dominating or the dominant. Particular trait is dominating itself in its expression. So that is what we call as That is, for example, heterozygous. You already know what is homozygous as well as heterozygous condition. If a particular gene is having a similar kind of allele, so that is called as homozygous allele. Right? A particular gene is having a pair of alleles, so that is of different kind, so that is what we call as heterozygous. So you can see an example here. Here it is of capital T. So this is of homozygous allele. So zygous of character type tall. That is called as homozygous tall, or also called as pure tall. So when it comes to the capital T as well as small t, this is also a tall, but it is a heterozygous. Next one, this is also homozygous condition, but it is a recessive one. Okay. So what are saying regarding the dominant rating in heterozygous condition? Okay. In heterozygous, where allele, right? In this heterozygous condition, the one allele will express. So here there is a capital T as well as small t. Here only the tall allele is expressing itself, and the dwarf gene is suppressed by the expression of this capital T. So that is what we call as dominant allele. That is, the one allele will express. So here, if there is a presence of capital T or the tall gene, it expresses itself. So that is the only one expression of this character, that tall, and this gene for a dwarf is not expressing because of the difficulty. That is what one of the that expressing, that is called as what? A dominant. Yes, capital. And the other one will be recessive. So the alleles which do not express itself, so that is what we call it as a recessive, right? So that is also called as recessive trait here. What is the dominant trait for height? That is also tall. What is the recessive trait? That is the dwarf. So which one is expressing here? That is the tall character. The tall trait is expressing. So that's why that is a dominant trait. The dwarf trait that is not expressing, that is what is called recessive trait. Right? So here it is a type of the part of type that physical characters of that is what is phenotype. Okay? It's genetic composition, genetic complement, or genetic makeup of product. Okay? Yes. So can easily identify, right? When you look at an organism, you can easily identify what is its phenotypic character by looking at what height it has, what size it has, what are the external characters, and external appearance of an organism, or physical appearance of an organism. So, how can you expect the genotype in the organism? It can't be seen with the bare eyes. So then, how we can go through the how we can know the genotype of an organism? Suppose this organism, for example, take an example of a tall plant, tall tree plant. So you cannot say its genotype by looking at it, right? Tall tree plant Either it may have the genotype of capital T, or it may have the genotype of capital T as well. Right. Which one you will get it? So it is a homozygous genotype, is a heterozygous condition. You cannot say it right, or by looking at the plant whether it is a homozygous or a heterozygous form. You need to test it. You have to test it with crossing method. So we are going to study the concept. Okay. Genotype of test it with the crossing method. So put it right. Fill up that the plant is tall, the plant is short. How it looks externally, that is what we call as genotype. So when it comes to the genetic composition, that is genotype, or the person can't see genes, and since you can't see genes, so that can be represented with cells. So that is a for homozygous cell, so that is for homozygous cell, that is one, one, that is height to the plant. So that is what. So the next part is about the Mendel experiment. You already know he is the father of Mendel. So before Mendel also, there are some scientists who studied genetics, but so the most experiments and scientific studies are this. That's why Gregor John Mendel is considered as the father of right. That Mendel's work has been recognized for his death itself. Period of time, his work has been recognized that after for a lot of years after his death, this what are the Mendel experiments? What experiments Mendel was? So Mendel was a priest in a church. He is basically a statistician. He is very good expert in statistics. So he applied those mathematics and statistical approach to study genetics. So he selected the key plants for his experiment and he conducted hybridization experiments. What are those hybridization experiments? You already know what is mechanical hybridization that you studied in the sexual reproduction in flowering plants. So what is that hybridization means or the crossing experiment means? So that hybridization involves what processes? It involves the emasculation. It involves bagging like that. So they will use selective breeding method. So they take the selective pollen and they will cross with the selective. So that is what we call as the hybridization experiment, where the Mendel conducted these experiments in a pea plant. So why he selected pea plants only? Because these pea plants can be grown in a short period of time. It has a very short lifespan. You can get the offspring in a very less period of time. That's why it has a short lifespan. That's why he selected one of the reasons. And so one of the successful features of Mendel's work is he got the pure lines. The previous work they didn't get how the pure lines are formed. The Mendel got pure lines. These pure lines can be made. So what are actually pure lines? Or also called as pure line breeding. This pure line breeding involves the continuous self-pollination within a plant over many generations. When a plants are self-pollinated for many generations, they will get a pure character. For example, on the tall plants, pure tall plants, homozygous tall plants. So what you have to do is you have to self-pollinate with the same plant for at least minimum 4 to 5 generations. Then you will get the pure lines that are tall or it will be for any character. Okay, got a large sample size, right? So very large sample. Whenever there is a large sample, you will get a different data. That is what that's right. Another one is contrasting character of contrasting traits. What are these contrasting traits? Opposing traits, for example, tall as well as dwarf. So then if you take the quad shape, so it may be wrinkled or one it may be inflated, right? Inflated or constructed. So like that, you got the contrast for different characters. For how many characters? You got 7 characters. For example, if you take some height. The height has a contrasting trait. What are those contrasting traits? Some height is a character. Contrasting traits are either it will be tall or either it may be short. Those are called as what? Traits. So we got 7 characters that has 14 or the pair of contrasting traits for each of the characters. So that is what he selected. Results better. So that's why we got what? Here the next one is of so what are those 7 contrasting traits? We can call it as 7 contrasting characters. Only 7 characters has a 14 contrasting traits. So that are these 7 characters are one is right, what is the the so character height equals for opposite traits. What are those? One is tall and tall. So you can see here the height that is one is tall, another one is dwarf. So that is a stem height character. So that is first character. The one is the quad shape. If you look at the quad shape, you can see here it is, here it is completely equal, right? Here it is one. And, so we can so that is what and they are indicated with the symbol. If the tall gene is the dominant gene here, that is indicated with capital T. So dwarf gene, the recessive gene, here when it comes to quad shape, the dominant one is here the dominant one is the recessive one, that is the right. So the quad shape is treated versus consistent. Dominant as in take the pod color. So some of the pea plants have a green pod and some of them have a yellow color pod. Among those, the green was the dominant one. That means that sample you got majority of them as a pod and some of them as a yellow color pod. So that is yellow is so that is another one is so I think you might have observed this plants they have different seed shapes. Some of the seeds are round and spherical. So some of them are wrinkled. So they have wrinkles on, right? Where round is the dominant one, wrinkled. So one color also different. Dominant seed color is so and the recessive seed color that is the one. So next one is that is the flower color. This flower color, the dominant one is the violet flower and the recessive one is the white colored flower. Okay. So come to the flower position. Okay. The flower may be axial, the flower may be terminal. So what is the axial terminal? The flower that will grow at axial position. Right. Here it is the flower grow. Flower six what? The terminal. right? So this is the term for character. So, the that that they as followed the how it is expressed. Whether it is tall or it is short. Whether it is yellow, either it is violet color flower or it may be the white color flower. Like that, that is the expression character in different forms. That are what we call the traits. How many traits are there? There are seven characters are there. So how many traits are there? There are traits. So the next part is you have to know what are pure lines. How you gonna get pure lines? Pure lines are also called as completely homozygous recessive. Whether if it is a tall, it is a homozygous pure tall. So if it is dwarf, it is homozygous recessive pure dwarf. So that is what we call as the pure lines. So how you gonna get pure lines? That is the self pollination is made through the crossing experiment over many generations, over at least 3 to 4 generations. You have to self pollinate them. The plant, okay, first they are completely they are either they are completely distributed one or they have some one. That is how the structure of pollination works. But what you will get right character. Then so you have taken a plant dwarf character. So you have to, but those are the plants which are having pure dwarf in nature. You have to self pollinate its own enters to the self pollinate. You have to self pollinate its own pollen grains with its own thickness surface. After a continuous self, you will get pure dwarf variety. So that is self pollinate. That the form to the continuous self pollination generation. Okay, like it self got pure lines for all those 14 traits. But 7 characters for all those 14 traits. He pure lines for all the 14 for for one is about us or crossing between one character is different. All the remaining characters in the 7 characters just the different for the class. All the other characters remain same equivalent character. That is what called as cross. Here you can see that all the characters, all the 7 characters, all the characters are kept same, but one character is kept. So what is that one character feature? From height, where he has taken one parent as a tall, and he has taken four dwarf varieties. That one character taken, and all the remaining characters kept in odd color, odd shape, green color, color, flower position. All the characters kept same. But only one character is different. That is a height character. He has taken one parent as a tall, parent one. So consider the parent will get a gamete. Parents are having a pair of alleles. Why it is a pair of alleles? You are having a diploid set of chromosomes, right? If you are having 26 chromosomes, half of from maternal side, half of from paternal side. So the diploid organism is having pair of alleles. That's why it is mentioned in the part of pair. But when it comes to the gamete, what happens? The gamete is in so it will reduce, right? It will reduce to a single cell, third cell. So the gametes are always haploid, so that's why cell it is the cell allele. So that is a capital T, capital T, parent or plant, but the gametes of eight gametes together, one all are tall, and all those tall plants were heterozygous. How you gonna know this heterozygous, whether it is a homozygous or heterozygous? Already you have taken what? Pure tall, pure dwarf. So then the gametes will combine, the gametes will fuse together, then what happens? Their alleles also combine. So that will be heterozygous tall plant. Then what he will do is he will cross the reverse of the self cross. So the artificial hybridization, self crossing, self pollinated them. Then what he got after selfing characters. But self then got the result is one all but a three tall plant and one is dwarf plant. Right. So he got a phenotypic ratio. What is the physical appearance of those plants? Three were tall and only one was dwarf. So then what might be the genotypic ratio? That is one plant is having homozygous tall, one plant is having homozygous dwarf plant. But how the genotypes can be known? To different types that is called as spectra, that is to identify what genotype was in the different but the class will be got tall and raw plant in the ratio one and the genotypic ratio is but again. So the Mendel, there are three different laws of Mendel, where the first law is that is the law of dominance, and the second law that is proposed by Mendel, that is the law of segregation. The last one is the law of development, where you can see the law of dominance, that is the first law. Right. So what this law states, that is when two different alleles are combined together. When they are combined, whenever the process is made, the two different alleles are combined. So among those, only one of them will be existent. What they are saying, when combine two characters, right? Here, already that is cross with cross link. What are the answers? What are the two different kinds are combining in a hybrid? So this is what they are saying. Two different kinds are combined together. All alleles of the cross allele, they are combined together in the hybrid. They are combined together. This what he got? F1 hybrid. He got all the tall plants in the F1 hybrid, right? So, so these two different alleles, that is capital T, those are combined together in only one of them is prescriptive hybrid. It got all the traits. The dwarf will be disappeared, right? The dwarf has been disappeared in the F1, but this dwarf has been reappeared in the F2 generation, right? So what we are according to the first of dominant, so only one of them will express. Which one is expressed? The dominant one is specific of two different kinds. But there are like what different alleles for hiding. So what is locus is position of the gene chromosome is called as locus. In this particular location, the allele capital T is located. So again, in another pair of chromosomes, there is allele that is not located on the normal allele. That is this normal allele produces normal enzyme. It expresses normally. For example, so as a DNA, the DNA is converted into RNA, then RNA is converted into a protein, and it expresses normally to take intent allele. This intent allele less efficient than the what? Less efficient. Less efficient expresses partially it will express it, or either it will not produce any protein, or it will not express itself, or it may have a defective expression, or it will have less efficient. So these cases are very rare where the plants or the organism get genetic diseases. But even though they are recessive, they are what? They produce are less efficient than them, but they are not as normal as as the normal allele. Now, here the wild variety, wild character with mutated, that mutated is defective or not is convert character together one head or other one will not exist. So we have this. This is about the law of dominance. So again, as already in the previous one in the monometric term, we got that in the F1 we got all tall plants, but you have to check it what type of alleles it has. Either it is whether it is homozygous or whether it is heterozygous. We have to know that. So we have to determine genotype. Then how we can know that genotype ratio is by making a test cross. So whether the plant which got in F1, whether it is homozygous or heterozygous, we have to test it. So how we gonna test it? So in the so in the F1 we got a tall plant. One got all the right. Even though they are all tall, but we don't know what genotype is. Because there are two genotypes, right? One is how we gonna test it? Tall plant, either it may be capital T or either it may be capital T, capital T, or right? Yes, the tall plant, you don't know that genotype, whether it may be that is unknown. So we cross them with, so this is the suspected plant. So we cross the suspected plant with a recessive parent, then it is called as what? Then it is called as test cross. So we cross this suspected plant with any of the parent, either the dominant parent or the recessive parent, then it is called as the background. So to determine the genotype of an F1 parent or to determine the genotype of an parent, what you have to do is you have to cross the unknown plant with the unknown genotype. Recessive parent, we got all were tall plants. So we got all tall. When there is not even a single dwarf, then this parent plant with the unknown genotype, capital capital right. So then only we got all tall. If there is, if we got any dwarf, then the genotype will be of heterozygous condition. Here, so if the parental plant, we don't know this genotype, if it has a heterozygous condition, we cross with this the parent, what you will get is half of them, right? Half of them you will get tall and half of them. So how this half comes? Because of the presence of a recessive gene, right? So it got from the both the parents, one from the parent and both parents must be capital. Tetra is track suspected plant with recessive parent, then it is called a test cross. We cross the unknown plant with any of the parent, either it is the dominant parent or of the recessive parent, then that is called as what? Back cross. Here itself, one example is taken, that is of a flower color. Here we don't know what is the genotype it has, whether it is homozygous or even it may be heterozygous. We don't know that, right? So what we're gonna do is we're gonna test it with a recessive parent. So it is of unknown genotype, whether it may be homozygous or even it may be heterozygous. We don't know that, right? So we cross them with parent. But all were violet plants, then definitely unknown flower is of homozygous dominant. Right. Again, the same thing. What we are doing is test cross. So we are always crossing the suspected plant with the recessive parent. Right. So if it is having a heterozygous condition, then this heterozygous condition we cross with recessive parent. If we cross with recessive parent, we get 50% of the flowers are violet and another 50% of the plants are white. If you got both the results, definitely it must be what? Two flowers represented. Right. So again, we will go back to the monometric form. Here, what we got? We got both heterozygous heterozygous the recessive here. So what we have to know is what is the F1 plant must be heterozygous because why it is heterozygous? We got all the type of characters, right? So if it is only homozygous, what might be the result? If it is only homozygous, we will get all the plants were again tall. But we got here the tall and dwarf we got. So that's why the F1 genotype is heterozygous. Imagine if we got F1 as capital T. So then if you cross with the recessive parent, again you will get all were tall, right? 100% is tall. But here, this case, some of them are dwarf. Even if there is a one dwarf line, definitely the parents are the F1. What? That is a heterozygous. Right. So that is what we call as the test term. So what is that? The test class, that is the crossing the suspected plant the recessive parent. Then you will get to know their type. That cross, that is the crossing the suspected plant the parent. What is the significance of this test class? The test class is how crossing with the parent. What is the what genotype of what genotype the plant. That is what the thing is in law, that is the law of isolation. Right? So what is happening here? So at the time of gamete formation, the time gamete formation the physical parent plant, the two alleles of a two alleles of during gamete formation, the two alleles of a gene is going to that the gamete carries one allele. So again, take an example of the same thing. Again, one more picture. Yes, so it is parent. The gamete has self right? Right. Self pollinate, yes, that is capital T. However, tall. So what happens during the gamete formation? What the alleles will separate? The dominant allele will separate, and the recessive allele will separate. They will form a haploid gamete, or they will come. Right. This is how the gametes are gonna separate during alleles are gonna separate during the gamete formation. Whether it is homozygous or whether it is what the definition is the gamete formation. The two alleles of a gene. That is what is the gene here? The gene coding for height. Height. What are the alleles here? It is tall allele. Another one is recessive dwarf allele. Right. So those gonna separate so that gamete carries one allele. So why the gametes are carrying only one allele? So that is because the gametes are having only haploid set of chromosomes, right? So they are having haploid set of chromosomes. So they will carry one allele. So when it comes to the organism character, that will carry a pair of allele. Why it is a pair of allele? Because it is a diploid cell. They have a so we have 23 pairs of chromosomes. Each pair of chromosome, it will, for example, if you take the first chromosome, that is gonna pair with the first chromosomes of the maternal side itself, right? So you should understand what are the homologous chromosomes, right? What are homologous chromosomes? So that are The chromosomes are having similar size, similar shape, so that's gonna pair. So that is what we call as homologous. Only so first chromosome always pairs with first chromosome only, right? It will not pair with 9 or 10 chromosomes or allosomes. So the first chromosome always pairs with first chromosome, maternal side, maternal chromosome, maternal chromosome. So that is what is one. So they will pair with their respective chromosomes, not with their chromosome, right? So here also the gamete carries one allele. So they are having only half set of chromosomes. So that is what the law of segregation is called as. So why? Because so this is also called as the law of purity of gametes. What happens? The gametes will be protected. That is during the gamete form. The two alleles of character, two alleles of the that are gonna separate. And in the gamete, there are only they carry only one allele in the gamete. The pair of alleles. So the pair of alleles is carried in the diploid cell or diploid organism. So this is the law of segregation. So the next part is uh, slight deviation from the Mendel's law. Mendel's as you have studied the law of dominance. So what it says, maybe one of the alleles will dominate and the other one will not be expressed. So that is what a law of one of the allele will be expressing itself and the other one will not. But there is a deviation from Mendel's law, that is what incomplete dominance. What is this incomplete dominance? So that one of the allele is not able to dominate completely. So another character, another intermediate trait will appear. That is what we call as the incomplete dominance. So what happening here? So progeny. So this progeny, that is consider F1 progeny, that doesn't look like what? That doesn't look like any of the parents. Suppose here, take an example. One example for this incomplete dominance is the snapdragon plant. The scientific snapdragon plant is, and another example for this incomplete dominance is Mirabilis jalapa, commonly called as. So take cross this snapdragon plant. So one is a red color flower, another one is a white color flower. So here the parents are color flower. Snapdragon plant is red color flower. Cross with carrot. Cross this will be the gamete that are on this, right. Cross them both. What is none of the parents will get a different. Doesn't look like it is parent. That is what that is. One what that is right. There that covers the character. Right. One here that indicates what? Dominant. There red color flower is or here the red color flower is on other character. Right. So that is third major character. That is the that is the F1 or what doesn't look like any of its parents. Here, the crossing is so that is a red flower plant, white flower plant. Those gametes are capital R as well as small r. So when they fuse together, what you get that is a F1 plant. The gametes, so what they do here, where the pollen grains, where the symbiotic activity, the ovary, where the gametes will reach, so then it will produce as well as the flower. So that plant is in the first plant. However, then so this F1 visit is selfing. If it is self-pollinated, so parental characters reappear in the F1. So that is one red color flower, two pink color flowers, seven flowers, and plant. That is so correct. One that what is that is the sixth one is concept of codominance. The name itself is codominance. That means cooperative dominance. So here more than one allele is dominating equally. Right. So that is the cooperative type of dominance. What happens in this case? Where both the alleles of the gene express themselves equally. Where only one is not dominating. Both of them are dominating together. So that is making cooperative dominance where both the alleles will express themselves in an equal way. So that is what we call as codominance. Here you can observe this codominance where one example is of ABO blood group is decided by the gene. What one of the specialties is ABO blood group is this ABO blood group has three alleles. Has what are those three alleles? A per one. So these are the three alleles that control the blood group of right. Here you get a different result where what happens here? So in a blood group where one is one allele is IA, another one allele is IB, then definitely the blood group must be AB blood group. So if it is having the allele only IA, it has an antigen, it has a sugar polymer on it. If it has the antigen A only, then the blood group must be A. So in case of IB, if the allele is IB, then the blood group must be having A, then the blood group is O. So if there are no sugar polymers, then the allele should be sugar, that is represented O blood group. So where is the codominance here? So the AB blood group where there are two alleles are expressing equally. So both are expressing. That's why it gives the blood group AB. But in the remaining, so there are only one allele. So here it will be dominance of AB. So here both are explaining. Both are expressing. Either it may be the same IA, or even it may be IA, or it may be here I will write all those possibilities in blood group IA as well as IA. Then this coming and but this that is having no antigen. It These are for A blood group. Then the thing to be for that, right? That is both the alleles is equal equally are decided by this that that right. That what both of the alleles express themselves equally, then it is called as what? Codominance. One of the examples of codominance is that is this video blood group is also example for well, that is what the multiple allele. The name itself is, for example, so all the characters have a we have a two set of chromosomes. So that's why we have a pair of alleles. But in case of this human blood group, the gene I has more than two alleles, right? Each and every pair of chromosome has pair of alleles. But here, case of human blood group, the gene I has more than two alleles. That is, one is IA and one is AB. These are the three differences that decide the blood group, different combinations of blood group. But out of these three alleles, only two can be found in an individual. Why? Because individual is required. This individual is required having pair of pair of more than two chromosomes. So each of them have a local only two of this. Either it may be IA or I. Either it may be IA or either it this only two alleles can be found in an individual because we are triploid. If we are triploid, then it might be more than two alleles. While all three can be seen at the population level. So some of them have IAIB, right? Some of them have only two of this. Some of them have only two of this. Some have only two of this. Or some of them have the same thing. Same allele, similar allele, IAIA or IB, IB. This is what two alleles can be found. Same, same, different. That is right. Here, so here the alleles from the parent one that is IE, so parent two that is IA. So that gives the blood group. If IA from the parent one, IA from the parent one. So if it is heterozygous A, then it is one parent process, that is IA, another parent process, allele, one I, that is heterozygous, that also gives the blood. Again, parent one may have the parent one may have IB, the parent two may have IA, again, you have to remember clearly. So what different blood group combinations are possible? So if you remember this that so that is easy. So what are the possible blood groups? So here it may be so A, B, C, and so that. These are all the possible blood groups. So this is from the maternal side. So from the paternal side, again the same possible group A, B, B as well as O. If you combine the paternal blood type, maternal blood type, so we means what happens? You will get a different blood group result. Father blood group is A, mother blood group is A. The possible blood groups are either A or even B. So how it is possible? So the other blood group that has IA and IE. Again, father blood group also has IE as well as IE. So that is the father blood. What are the possible blood groups here? A and A combined, it will be A and I combined. Okay, I and I combined, what it will be? Only possible blood groups in the offspring, that is of A or like that. So even if our father has B, mother has B, all possible blood groups are for father's blood group. So these are the possible genotypes. As part of this, then please remember what type of parental blood group is what type of blood group. So this is all of the multiple allelism. So example for multiple allelism. So in some case in the human blood group, so that is controlled by the gene I has three different alleles, more than two alleles. What are those alleles? IA, IB, and the I. Those are the alleles that are responsible for multiple allelism. So the next concept is of pleiotropy. So what is this pleiotropy then? So when it comes to the pleiotropy, we can see that there what it defines is where a single gene controls any character. That means only one gene is responsible for expressing any characters in the organ. More than one character in the organ. Then it is called as what? The pleiotropy. What are the examples for pleiotropy? That is a starch synthesis in species. Another common popular example is the benign fetal. Right. First one study how only one gene is controlling the characters in the sequence. So that is a starch synthesis. Right. If the more starch is synthesized, the size of the seed is increased. Right. So here the gene capital B, alleles capital B as well as capital B. So here the seed shape is round as well as size is large. What characters are controlled here? That they are very. So if it is a homozygous recessive, the shape must be wrinkled and the seeds are small. So this is a recessive character. If it is dominant, it is round shape as well as large seeds are observed. If it is of capital B and small b, it is a heterozygous condition where seeds are round and the size is intermediate size. If the size is intermediate, again, what where we see this intermediate expression of character? The incomplete dominance, where we got pink color flowers, that is an example for incomplete dominance. Here also, the pleiotropy in seeds, where it is a starch, where you can observe the intermediate size of the seed, where they show what? They show incomplete dominance for a size only. Again, the shape is round, but the size is varying. So, what is the size we get in case of heterozygous condition? That is of intermediate size. Neither too small nor too large. We will get intermediate size that shows incomplete dominance. physiotropy. For sharp. Another example for physiotropy, that is called as phenylketonuria. This phenylketonuria is a inborn error of metabolism. So that means by birth itself the disease comes, and the death is inevitable. That is inborn error of metabolism. What metabolism is affected here? And this disease is an example for of where it is a autosomal recessive disorder, where you can see this, where this phenylketonuria, where the phenylalanine is converted into a tyrosine in the presence of an enzyme phenylalanine hydroxylase. So this is the normal case. In the normal human being, where phenylalanine, what is this phenylalanine? It is a amino acid, right? So this amino acid, that is phenylalanine, is converted into a tyrosine with the help of the enzyme phenylalanine hydroxylase. If the person is affected by phenylketonuria, what happens? That person acts an enzyme, that is phenylalanine hydroxylase enzyme. This phenylalanine hydroxylase enzyme is lacking in a person. What happens? This phenylalanine is not converted into a tyrosine. If it is not converted into a tyrosine, what happens? If this phenylalanine is converted into what? Phenyl pyruvate, because phenylalanine hydroxylase enzyme is absent. Then phenylalanine converts to phenyl pyruvate, and this phenyl pyruvate starts to accumulate in the brain. So when it starts to accumulate in the brain, so it finally leads what? Mental retardation by birth itself, because it is inborn error of metabolism. So this is what one character it is saying. Again here the skin color is affected, and again this phenyl pyruvate is not fully absorbed by the body because of the poor absorption, poor reabsorption by the body. So this can be excreted. Phenyl pyruvate can be excreted in the urine. That's by the phenylketonuria. Right. So here it is a structure of the phenyl. The organic compound, where one carbon group is there, amino group is there, acid group is there, alkyl group is also there, along with, right. If the hydroxylase enzyme is present, hydroxylase, then the hydroxyl group added, then it is converted into another amino acid. If this enzyme is lacking, it leads to the error of metabolism, where the phenylalanine is not converted into tyrosine. In turn, it is converted to phenyl pyruvate that accumulates in the brain, causes mental retardation. Because of its poor absorption, it can be excreted in the urine, and the later stages they also lead to the failure of the. So this is what one other example for what? Pleiotropy, that is single gene. The gene is coding for the enzyme phenylalanine hydroxylase. It is controlling many characters. What it is causing? Mental retardation. It is also affecting kidneys. It is also affecting skin color. So those are many characters that are controlled. That is what the definition is. Single gene control character. That is what we have. So, example, starch synthesis. Plant species. Another example is, yeah, what? So the next one is of a dihybrid cross. As the name itself says, it is a dihybrid class. So first we recall what is monohybrid class. So in the monohybrid class, what we did is all the seven characters, among the seven characters, 6 characters kept the same. Only one character is taken of a different possibility. That is, one plant is tall, one plant is dwarf. This means what we are going to do is, there is We are we are experimenting on inheritance of two characters where all the other characters are kept same. The two characters are taken, the different possibilities, probabilities. All the remaining were kept same. What are those two characters taken? That is of seed shape as well as seed color. That is round and yellow seeds are taken. And this residual part that is wrinkled and green. Again, seed color are taken in the parent. Here, the two characters are taken different. All the remaining characters, stem height, and pod shape, pod color, flower color, flower shape, that same. Only two characters are taken differently. One is round shape, yellow color shape, another one is wrinkle shape, green color shapes are taken as a parent. These are taken as the parent. So, for the parents, one is the taken for yellow. Then, right, what the here, but those are what different of the gametes. What are the what are the we are going to get different combinations of two generations. Right. What here left one is going to be round yellow. That what will be what what will appear in the F2. So this is the representation of dihybrid class where in this dihybrid class what they made, what they have taken. So you can see that. So they have taken a round yellow seed that is the genotype capital R, capital Y that are crossed with the wrinkled grain that is small r as well as the small y. So in the gamut get capital R, capital Y. So they fuse together. You will get what? That is the F, okay. One generation pollinated. You will get different gametes of this capital R, capital Y, capital Y, capital R, small y. both really have together. So what then? Who gonna get this? This is the ratio. That is the capital R, capital R, capital. So what is the shape here? It has the color. Where you will get round yellow. Right? Again, if you cross with capital R, capital Y with small r, capital Y, again you will get yellow. This here also is like that. You have to cross all this side. Okay. Sorry, there is a part. There are two different characters as we see. The two general will get one. How this picture has been got? That is, here we have taken two characters, right? So in the mentality of 3 to 1. So it is multiple in the diagram graph. So it is can what are those round yellow seeds? All these will be the what are those round seeds where you will get the round colored yellow. So that is a where it is called yellow. So we will take the genotypic ratio also. In the genotypic ratio also, what is the genotypic ratio of monohybrid plant? So here you can see 1 is to 2 is to the size it is a dihybrid plant of 1 are having dihybrid plant where the two characters are taken different characters, all the remaining characters, all the remaining 5 characters get the same. So the next one is the law of independent development. The name itself says what happens here when the two characters are combined. So what happens when the dihybrid cells, two characters are combined? One is red, one is yellow. These two characters are combined to hybrid. So this is what hybrid, right? It is down the row of heterozygous cells. So this is F1 generation of dihybrid cells. So what we are saying when two characters are combined in a hybrid. Therefore, their traits can segregate. Their traits. What are the traits? Round yellow and then red green. Other recombinations also appear. So what are the main parental types? There are only two main parental types. One is that is a dominant type. What is that? That is the round yellow. Round, round is as a yellow color. Another one, recessive parent, that is a what is the definition? When two characters are combined in a hybrid, these two characters are their traits can segregate and they will be combined in any combination possible. Apart from this parental type, we also got different possibilities. What are those different possibilities? One is the round green, the one is third, third. Okay. Even in case of genotype also, one is to two is to one. That are what all the different genotypes got. There are different genotypes also. That is what different characters are combined in a hybrid. So that are gonna recombine in a different combination possible. That gives us that makes every possible combination to be F2 generation. That is what the law of independent. So the next concept is of the polygenic inheritance. The name itself says poly means poly means poly, yeah, are controlling character where we are more control the expression of same character. So it is exactly opposite to pleiotropy, right? What happens in pleiotropy? Single gene controls many character, but here many genes control same character. That is exactly opposite to pleiotropy, right? So where the three or more genes control expression of a same character, where you can see, for example, skin color as well as height. So that is what gradient. So one example for polygenic maintains the skin color. Skin color is controlled by three different genes. What are those three different genes? These three different control the character of skin color. How it controls we can see. Where if all the genes are dominant, all the three genes are dominant. So that gives the darkest skin color. More melanin pigment, that is the darkest skin color. If all those genes are of in a heterozygous condition, capital A small a, capital B small b, capital C small c, that gives rise to a intermediate skin coloration. And if it is of all are recessive, small a, small a, small b, small c, that gives the lightest and brightest skin color. And these genes have a additive effect. What is that additive effect? If more dominant genes are present, that adds to the coloration. That gives the darkest color. If there is a less dominant genes are present, it gives the lightest color. So that is what we call as additive effect. If more dominant genes are present, there that gives darkest color. That darkest color. So the next part is of some of the questions. We will take some of the questions from the all the covered concepts. So the first one, the question says, given below are the two statements where the statement 1 says the Mendel studied 7 pairs of tree and proposed the law of inheritance. He proposed the law of inheritance. Is it true or is it the correct statement or a incorrect statement? Yes, Mendel studied 7 pairs of contrasting traits. 7 pairs, 14 contrasting traits for a 7 characters. Yes, is right. So we in what plan? Studied in a plan, and based on that result he proposed the laws of inheritance, law of segregation, law of all this. So the second statement says these 7 characters are examined by Mendel in his experiment on a pea plant. Where, so what are those 7 characters? One is the seed shape, yes, seed color is there, flower color is there, pod shape is there, pod color is there, flower position, stem height, tall and dwarf, seed shape, wrinkled and round, color yellow and green, flower color violet and white, pod shape that is inflated and consistent, pod color again green and yellow. So all these were the 7 characters that is statement. So in light of the above statements, who is the correct answer from the options given below? Here, the first one, the statement 1 is incorrect. They are saying is wrong. Statement 1 is also correct. Statement 2 is also correct. So the correct option to this is both statement 1 are correct. So they studied 14 contrasting traits or 7 contrasting 7 characters. 14 or 7 pairs of contrasting traits. Both the statements are correct. So the next question, so here the production of gametes by the parent, production of gametes, formation of zygote, the F1 and F2 plants can be understood from a diagram called as. So what is the diagram? You put the square diagram you put while crossing F1 and F2 generation. That is called as what? The Punnett square. What is this Punnett square? It is graphical representation where the generation F1 generation and F2 generation is studied in detail with all the possible combinations. So that graphical representation is called as what? The Punnett square. So the next question is identify the wrong statement with respect to the gene I. What is this gene I? To control blood group. Blood group has will control. What you have to do is you have to identify the statement. So a person will have only 2 of this allele. Right or wrong? So yes, there are different alleles are there, IA, IB as well as I. But if a person can only have the 2 alleles but not all the 3, so the statement is right. Only 2 of the alleles, only 2 of the 3 alleles are present in a person. But at the population level, all the 3 are present at the population level. Right. So the first statement is right. What you have to identify? You have to identify again. When IA and IB are present together, they express the same type of sugar. Right or wrong? They express the same type of sugar. Is it true? The A group has antigen A. The B group has antigen B. What they are saying, when IA and IB are present together, AB blood group, they express same type of sugar. Is it true or false? Definitely it is a false statement. But A has a A antigen, B has B antigen. They have a different sugar polymer. They can't have the same sugar, same type of sugar polymer, or same type of antigen. Right. So the allele I, so that's the wrong statement. The allele I does not produce any sugar. Yes, it does not produce any sugar. Right. So it means the small I. The gene I has three alleles. Yes, definitely it has three alleles. What are those three different alleles? IA, IB, or what? The different types of alleles for I, that is one. That is how many true breeding, how many true breeding, that is learning. So it self select, but it has except in one character, except said earlier, pure line, they are purely homozygous, produced through the pure line breeding by a continuous self pollination. What Mendel did was, so there are 14 contrasting traits. So all the 14 traits, Mendel got a pure line. He produced pure line from all those 14 traits, and he also crossed them by the monohydrid crossing. So this one is a 14 traits. The two breeding plant varieties right? They are not asking the pair. They are asking how many seed plant varieties. So 14. It's a 14 different traits. Which were similar except in one character. That means for a monohybrid, so he produced 14 pure line Mendel failure. Where did the Mendel fail then? That is, he used mathematics and statistics in the biological concept. Mathematics has that statistical application in the biological method that makes it complicated for the scientists to understand what Mendel was trying to. So that is, he used mathematical statistics for the biological biology also. Right. The next one is the publicity. He got a very less publicity for his work. Publicity of the Mendel's work was here. Nobody could understand what Mendel did in this experiment because he was ahead of his time. Nobody can what he did was. Means the Mendel's work was ahead of his time. What is the unfortunate thing is, so the Mendel's work has been started rediscovering after his death only, right? So father of genetics is the Mendel, but Mendel did not know what he is studying genetics. He doesn't even know what is gene. He called them as factors, right? In the law of dominance, so that he called the genes as a factor. Later, that was known as genes in the later time after his death, right? So where did Mendel fail? Is communication, publicity was very poor. Then the mathematical and statistical principles applied in biological concepts. So that makes it complicated. And you at that period, at that time, nobody could understand what Mendel is trying to say. The next one is Mendel's work was ahead of his time. He was thinking ahead of others, other of his contemporaries. That is of Buddha. So this Mendel's work was rediscovered by three different scientists: Vidyapeeth, Carl Correns, and von Sherman were the three scientists. During the 1900s, they rediscovered the Mendel the during and so the next one is the chromosomal theory of inheritance. So as the name itself says, chromosomal theory of inheritance. So according to this theory, inheritance, that is the process of transfer of characters from a parent to offspring, that was connected to the chromosome. So the name itself, the concept itself is chromosomal theory of inheritance. This chromosomal theory of inheritance was proposed by the scientists Walter Sutton, Theodore Boveri in the year 1900. According to this theory proposed by Sutton and Boveri, what they said is the information of what? Information, or you can consider the genetic information or information of organism, all its characters, all its phenotypes, all its abilities, all the information of an organism that pass from one generation to the next generation through what? Through the chromosome. So, the Sutton and Boveri called the chromosomes as vehicles of inheritance. What is the vehicles do? They transfer, they transport, right? So, that is, they called them as vehicles of inheritance. So, what they carry? They carry the information from the parental generation to the offspring. From one generation to the next generation, they pass the information. They carry this inheritance. So, that is carried through what? Carried through this chromosome. The information, all these information present on the chromosome. And we also noted that the behavior of chromosomes was parallel to the behavior of genes. So genes as well as chromosomes, they behave like a similar pattern. What they observed is chromosomes as well as genes, they behave in a similar manner. So what they observed between similarities between the chromosomes and genes was both occurs in phase. So here you can see the gene coding for capital T, that also occur in phase. Again, if you take chromosome pairs, there are 23 pairs. Why we call it as 23 pairs? Because they occur in pairs, right? First chromosome pair with first paternal chromosome pairs with first paternal chromosome, right? They also what? Occur in pairs. Both genes as well as chromosomes occur in pairs. What is the next one? Both of them segregate at the time of gamete. Yes, definitely. These genes also separate. These chromosomes also separate at the time of gametogenesis, at the time of gamete formation. Here, what? The gene coding for height is located on the same locus, both on maternal chromosome as well as maternal chromosome. Either it may be capital T, either it may be capital T, or one may be small. Any of them. So they are located on the same locus, each pair of chromosomes. Right? Yes, both of them segregate the time of gamete formation, such that only one of each pair is transmitted to the gamete. Why only one of them is transmitted to the gamete? Because again they have to combine in a fusion, right? When the fusion takes place, again the gametes will combine. Again they occur in pairs. Again when it comes to the chromosomes also, the haploid set consists of 23 pairs. Again when the fusion takes place, again they become 23 pairs. Right? See, one of the each pair is transmitted to the gamete. The independent pairs segregate dependent pair. Again, one pair of chromosomes is not dependent on the next pair. They segregate dependently of each other. Dependent pair, this is one dependent. That is another chromosome is of another independent pair. If we take allosomes also. So, the independent pairs segregate, separate independently of each other. So that is not connected, that is not dependent on any other pair. This is what the similarity observed by the Sutton and Boveri in both genes as well as this chromosomal theory was proposed by whom? Sutton and Boveri in their. According to this theory, what it is? Chromosomes are the vehicles of inheritance. That is, information passes from one generation to another generation. That is via the chromosome. Through the chromosome. That is what the theory is called chromosomal theory of inheritance. But the experimental proof of this theory was given by another scientist. That is the pH organ, Thomas organ, is father of this problem called as part of experimental genetics. So that is experimental verification of chromosomal theory was made by PH1. So what organisms were used by the PH1? He used the Drosophila melanogaster, right? He used Drosophila melanogaster. What is why he used Drosophila melanogaster? So because of the use of Drosophila melanogaster, PH Morgan got the Flyman of Genetic. Yes, because of his experiments on the fruit fly, that is Drosophila melanogaster. Why he used Drosophila melanogaster? These can be grown in a simple synthetic media, culture media, in a laboratory condition. In a short period of time, that can be synthesized in a laboratory. Right? This is the one reason he used Drosophila. Fruit flies for proving the experimental verification of. Again, why? Because this Drosophila has a very short life cycle. About 2 weeks it will complete its life cycle. From the day 1 to day 14, it completes its life cycle. So all the juvenile stage, the reproductive stage, and all those stages are completed just 2 weeks. Like they reproduce in a typical table, they will meet and they, the single meeting should produce large number of progeny. Right? Single meeting of a population that will produce large number of offspring. 2 weeks, within a 2 weeks, within 1 week, you will get that progeny slide. Again, here there is a clear differentiation of sexes are also there, right? The male is smaller than the female. The female has large abdominal strip. Male has a smaller strip. Then the male has a sex comb in their toe, in their spots in their feet. Then next one is the frontal limb. They have the sex combs that can be easily identified. They are present only in case of male. So, in case of female, the females are very small than the male. Abdominal strip. Those are the differentiations between male and female fruit fly. So then the next one is the many type of hereditary variations can be seen. So we observe many different types of hereditary variations. The variations created in the Drosophila. So what are those? So some are red-eyed, white-bodied, right? Some are large, wings, and some variations in the organisms. Even under the low power microscopic. That is why he selected for this experiment. So the next one is of physical association of chromosomes. So that is what we call as the linkage as well as recombination. So when it comes to the linkage and recombination, it is the next concept is of linkage as well as study. First, what is this linkage then? For example, if you take the chromosome, so the genes are located on a chromosome, right? Are located chromosome specific positions that are called locus, are also called as locus. So we have a particular gene for eye color. So eye color, eye color. So we say that locus. status. Yes, here also again there is another gene, another kind. If they are in a close proximity, if those genes are close proximity, so they are associated with one another. The recombination possibility of a recombination, possibility of a crossing over is less when they are associated closely on what is they are saying the linkage is yes, physical association with genes in a chromosome. If the genes are associated closely on a chromosome, they tend to uh, their tendency of separation, their tendency of the exchange of chromosomes, even the exchange of chromosomes segment takes place, they remain same. The frequency of recombination is less when they are closely associated, when they are closely located on a since they are located on a different position. Again, the same is taken example. They are located at different distance where more recombination is possible here. That's the concept. When the genes are closely linked, are closely linked, the frequency of the genes are distantly located. The frequency is more. Again, the next one is what is recombination. Already we studied in the genetic concept. What is crossing over? What is recombination? What is crossing over? It is the exchange of chromosomes on sister chromosomes. What crossing over that leads to what? You will get combinations that are not same. Get variations within the that is what we call that. What Morgan observed. Morgan observed when the two genes in a dihybrid cross were situated on the same chromosome. The two genes, this is one, the two genes in a dihybrid cross were situated on the same chromosome. The proportion of parental genes, the proportion of parental gene combination was much higher than the non-parental type. Parental even though dihybrid cross takes place, even after the crossing, the variations are less. They are almost similar to their parents when they are located situated on the same chromosome and closely linked. So, when the non-parental appear, non-parental combinations are possible when they are located on a different chromosome. They are located on different chromosomes then the non-chromosome possible is higher. So, two genes dihybrid cross situated on a same chromosome, the proportion of parental gene combination again appear is more higher than the non-parental type. So when the non-parental appear then if they are located on a different chromosome, when they are not closely linked, then the recombination is possible, the proportion is more possible. Adding to this, linkage is inversely proportional to right? The linkage is more, recombination is less. The recombination is more, then the linkage is more, the linkage is inversely proportional. what? The recombination. That is what about the linkage. So, the physical association of a gene and a chromosome is called as linkage. Recombination, that is the generation of parental genes. When the chromosomes recombine, they generate non-parental characters, new characters, right? So that is what called as the recombination. So the next part is about the linkage mapping, or also called as genetic base mapping. So this concept of genetic mapping or a linkage mapping is given by the Alfred Overton, who was one of the students of P.H. Morgan. So what he used, this Overton used the frequency of recombination between the gene pairs and the same chromosome as a measure of distance between the genes. He used frequency of recombination as a measure of distance and mapped their positions in the chromosome. That distance is measured in centimorgan, also called as gain. Here, what he did was, very what he observed was of from here, what as a measure of distance between they are having the less called as what? For example, 1 centimeter, very less recombination, right? So if the frequency of recombination is more, the frequency of there is higher here. So here the frequency less if they are located or closely connected. That is the distance between those two are for that. Here, so in the diagram you can see a homologous pair of chromosomes. So this is when the crossing over takes place, and these are the gametes of the recombinant. Right. So this is the homologous pair where the A and B genes are located here, right? So they are closely linked. So the linkage is more here. The physical association between the genes on a chromosome is more between A and B here, right? But between A and B, they are more close. Between A and C, they are distant. They are distantly located. Only linked genes, they are distantly located. Right? The frequency of recombination is more between A and C, but where it is less between A and B. So the frequency of recombination is less between the genes A and B, but A and C there is more frequency there. So it is a crossing between performance paternal are that will crossover. The crossover, there is an exchange of chromosomal segments, maternal and paternal. When they exchange, genes also are located on those segments. So in the recombinant, what happens? So here the C from the maternal chromosome comes here, the C from the paternal goes here. Again, but this closely linked genes, they will not recombine. They will not vary. They will remain as it is. So when they are closely associated, when the genes are closely associated, their recombination is very less. The frequency of recombination between linked genes is less. When they are distantly located, there is a lot of possibility of recombination. So that is what it is. Right? The distance is measured in terms of what? 1 centimeter. The frequency is less than 1%. The frequency of recombination is please inversely proportional to what? Recombination. More linkage, here it is, more linkage, but the recombination is less. So here it is, more distance, so that's why linkage is less, so the recombination is more here. Recombination is high here. So that is what the point is. That is how it is connected to recombination. So Morgan did experiments on Drosophila melanogaster to study what? To study sex-linked genes or X-linked genes. What he did was he made two different crossing experiments. So in the first crossing, he considered the characters, that is wild characters, that is brown bodied and red eyed. He considered a dihybrid cross. He has taken two characters. One is having a brown body and they are having red eye. Right? They are having a red eye. Here Y plus that indicates brown body. W plus that indicates red eye. So here it's mutant character. The recessive character, that is a yellow bodied and a white eyed female. That is small by small. So what they say is, in this first crossing, these gene coding for brown body, red eye, they are in a close proximity with each other, since the body color and the eye color genes are closely linked. When they are closely linked, the recombination is less. Variations will not occur more than this. That means they are similar to parents. They are similar to parental combination. Similar to parents. How much? 90. 7% they are similar to their parents. Only 1.3% variation. Only 1.3% recombination has taken place in this crossing. But in the second crossing, here it is red eyed. He has taken eye color as well as wing size. Red eyed Drosophila and large winged male Drosophila. That is of the wild varieties. So he crossed with a mutant character, that is white eyed as well as smaller wings, miniature wings. When these are crossed, where the genes are loosely linked, so the recombination, the frequency of recombination is how much? 37.2% recombination frequency, where the genes are loosely linked. So the diagrammatic representation is already given in the NCERT text system. Here you can see that. So in the cross one, here, yellow bodied, white eye. So this is what the wild type of it. Here, so in the F1 generation, they got similar. There is no recombination here, no recombination here. White. So recombinant. So how many? What is the frequency of recombination? 1.3%. So 98% almost similar to what? similar to the parental type. So you can see here, this one and this one. Okay, this one. But in the other case, where he has taken white-eyed meaning wild type, where parental type is 62%, frequency of combination 37.2% taken is the what loosely linked. Here also you can observe what is the position of genes here. You can see that. Yeah. Okay. Here it is. One thing, here it is. But here you can see that they are closely linked. Here also they are closely linked here, but they are distinct here. That is what that causes the frequency of recombination. They are loosely linked, frequency of recombination is high. They are closely linked, frequency of recombination is only 1. Right. So that is what is the recombination organism. So the next part is about the sex determination. So sex determination. So sex is determined by either by male or by female. Sex determination is first traced by the scientist in Henkin. Right. This Henkin in the year 1891 he traced a specific nuclear structure to the spermatogenesis in insects, called it as the X cell. What he studied the sperms of the insect, and what he observed as he called those sex chromosomes as X bodies. Right. So that was later identified as what X chromosomes, which are determining sex of the insect. Right. He studied insect. Then what are the types of sex determination? There are 3 different types of sex determination. First, XO type, then Y type, then ZW type. Here it is XX XO type. Here it is XX XY type. Here it is ZZ ZW type. So this XX and XO type is observed in case of grasshopper, and Z and ZW is observed in case of birds. XX and XY type it is observed in case of grasshopper. So what happens to sex? So the males have only one explanation, the one and so 2 chromosomes. Right? So the male has only one chromosome. Another sex is absent, and the female has 2 X. Like grasshopper. But in case of female, XX chromosomes are present in case of female. So here it is called as male heterogamity. So male determines the sex of an offspring. Right? So that is what males have the XY, males have. We will study how the chromosomal pattern goes. Next is ZW type, that is observed in case of birds. Here, the females are the heterogamous. Females determine the sex of the offspring. Heterogametic. Hetero means different. Gamete, different gamete. The females have Z as well as W. The females are homogametic. Same type of gamete as ZZ. So that is observed in case of sex. We will see how the pattern works. In case of human beings, we will see XX chromosome is for male allosomal pair, XY is present in case of male. So how many chromosomes are there in a human cell? There are 46 chromosomes. Among these 46 chromosomes, 44 chromosomes are called as autosomes. Autosomes, somatic chromosomes, body chromosomes, right? That are common in male and female are called as what? Autosomes. They are both common in case of male and female, but only difference where it comes. X and Y are called as allosomes. are also called as sex chromosomes. Right? So these allosomes are sex chromosomes. Which type? So in case of human male, there is Y chromosome. In case of female, there are two X chromosomes. Allosomes. So this is the here you can see the autosomes are 44 plus XY is the chromosomal combination. So here it is. The male is heterogametic. So the gametes consisting of 22 autosomes, 1 X chromosome. Another gamete consisting of 22 autosomes as well as 1 Y chromosome. That is a heterogametic condition. So this in case of females it is 44 plus 2 XX chromosomes. When it comes to the gametes, there is 22 autosomes with 1 X chromosome. Another one is of 22 with another X chromosome. And this combines with Y gives rise to this combines with another X chromosome which gives rise to organism. But when it comes to the insects, here also the same thing. X here also it is female homogamity. Here also it is male having only one X chromosome and one is null chromosome. This XO indicates no chromosome there, only having one X chromosome in case of insect. Right? What happens when X chromosome is present? Some of the male gametes have X chromosome, some of the male gametes have no chromosome. So here also, this also indicates determines the sex of the offspring. You will see how the sex determination takes place in different types of organisms. So sex determination mechanism in case of insects and grasshopper is of XX and XO type. So here XX is of male, XO type is of XX is of female, XO type is of male. The male is heterogametic, which determines the sex of the offspring. So female is having two X chromosomes here. So where it is observed? It is observed in case of insects, particularly in case of grasshopper. So XX, XY, here also it is male heterogametic. So here it is XX chromosomes are in female, Y is in case of male. So it is observed in case of human beings as well as grasshopper, where the sex is determined by the male organism, male individual system. Right, here it is same in case of female also. Another one is ZW mechanism. ZZ. So here in case of birds, sex is determined by this female individual, which is heterogametic. Two different types of gametes. This when it is combined with one another. Whenever it gets ZZ, it is becoming. Whenever it gets ZW, it becomes a male. So that is observed in case of what? In case of male. In case of bird. Next one is haplodiploid type of sex determination. What is this haplodiploid type of sex determination? So here it is. The males are haploid, females are always diploid. Here is male. Whoever diploid, not diploid. Here it is observed. It is observed in case of honeybees. Right? So the males are drones. Already I have seen the social organization honeybees. There are queens, there are workers, there are drones. The queens are always female. The workers as well as drones are workers are almost female. The drones are the only males. They are having only haploid gametes, only haploid set of chromosomes. Whenever they have only half number of chromosomes, they become males. Whenever they have two sets of chromosomes, then they become what? Then they become a female. Either they become queen or they become a worker. So that is observed in case of queen. There you can see that the queen is always diploid. So even the workers are also diploid. They are having 32 chromosomes. Here in case of drone, they are having only 16 chromosomes, they are always haploid. So we can study, we can elaborate more the sex determination in fertility. Here you can see, as I said, female is always diploid consisting of 32 chromosomes, right? It is always consisting of 32 chromosomes. When it comes to the male, it is having only 16 chromosomes. What happens is, female produces the gametes through the process of meiosis, it will produce haploid gametes. And so these two haploid gametes, when one female gamete fuses with the male organism, so this is a drone which undergo mitosis, again it will produce what? Male gamete. So this male and female gamete will combine. The fertilization takes place, it becomes female. When the parthenogenesis occurs, the gamete produced by the female, this will convert it into a male organism. What is parthenogenesis here? That is, the organisms which are formed without fertilization, those are called as what? Parthenogenic organisms. Where without fertilization, this male gamete becomes male organism consisting of the chromosomes. Right. So the next one is of, so this is of the sex determination in case of animals. So the next one is of the mutation. Right. So what is these mutations is all about? So when it comes to the mutation, so it is any alteration or any change in the DNA or change in any point of the chromosome is called as what? The mutation. So when it comes to the mutation, it is a phenomenon which results in alteration of a DNA. So it changes the normal DNA. So that is what is called as the alteration of a DNA, and it consequently results. So that is what we call it as the mutations. So when it comes to this, there are different types of mutations. Among those, the first one was the point mutation, and the another one was the frame shift mutation. Again, these point mutation as well as the frame shift mutation was caused by different processes, different mutations called as the deletion, called as the deletion. Another one is repetition. Another one is insert. Suppose if you take a particular DNA segment, so that is of A, A. So this is a particular segment of DNA where, in case of deletion, some of the nucleotides are deleted from the segment of DNA. So that is called as deletion. Here, this part GC has been deleted or AT has been deleted. So that is called as what? Deletion. That also causes changes in the DNA. Next one is called as repetition. What is that repetition? The repetition of the same segments again and again. That also changes or alters the DNA. That is also called as what? Repetition. Another one was the insertion. Insert. That means some of the other segments has been inserted in a normal DNA segment. So that is called as what? Insertion, where normal segment is ATGC, AC and AC. So what happens here? Along with ATGC, the GA has been inserted. So this also alters. So that is called as what? Insertion. So the mutations occur due to deletion, or might be repetition, or might be insertion, that alters the DNA sequences that if the DNA sequence is altered, what happens? When it transcripts, RNA also alters. When it translates, again the protein also alters. So whole expression of phenotype as well as genotypes are altered here. So that is what the mutation causes. There are different types of mutation. First one is point mutation, another one is range shift mutation. So what happens in case of the point mutation? So in case of point mutation, there is a change in any single point where only single base pair has been deleted or single base pair has been changed. So that is what we call as point mutation. Example for a point mutation, that is a sickle cell anemia. So we are going to study in detail the point mutation as well as frame shift. So when the frame shift mutation is caused, if there is a deletion of a base pair or if there is an insertion of a base pair, what happens? Suppose a particular triplet codon is coding for an amino acid. So if it is changed due to deletion or insertion, what happens? The genetic codes will change. So the triplet codon code for different amino acids. So if the different amino acids are there, then the expression will be different, which also causes the genetic disorder. What is the cause behind this mutation? Mutation can be caused by the substances called as mutagens. So mutagens, these are any chemical substances or the physical factors that induce, that cause mutation are called as mutagens. Either it may be harmful radiation like X rays, gamma rays, UV rays. So those are called causing what? Different types of mutation in the organism. Right. So the first point we are going to study is of a point mutation. These are the base pairs A, C, T, A, as well as suppose at this particular point that has been changed. What happens? So instead of thymine, the cytosine will appear. What happens? The whole expression of this, the whole translation, the expression of a phenotype changes due to a change in only one base pair, change in only one point of the DNA sequence that alters the whole character itself. So that is what we call as point mutation. So one example that we are going to study, that is of cell anemia. So what happens in cell anemia? So we will study the genetic disorder, which is a result, which is an example for what? Point mutation. So where the RBCs are generally the round, disc shaped and biconcave structure. When the person is affected with the autosomal recessive disorder, there is a sickle cell anemia. What happens here? The RBCs get sickle shaped. Surface area of the RBCs reduces. Oxygen tension is very less. So that is going to happen due to what? Due to this point mutation. So we will study this in detail with genetic disease. So the next example is that is of a frame shift mutation. So what they are seeing, the name itself is frame shift mutation. What happens here? See, this is what the normal sequence where A, T, G, A, G, E, C, this is a particular sequence of a DNA. So where what happened here? Due to insertion after a A, T, A, G, T, but here it is A, T, G, A, E, T. So here another frame of a base pair, another base pair has been inserted due to the errors in the process of in the errors in the process or the phenomenon. So that leads to what? Insertion of a particular base pair. If this base pair is inserted, see here, this ATG, so that transcripts into UAC, where DNA becomes RNA, so A becomes A, C. And this ATG codes for the amino acid that is of tyrosine. AGT is coding for amino acid serine. And CCA is coding for the amino acid lysine. And AGT is coding for the amino acid that is again serine. What happens if the insertion of another base pair here ATG code for tyrosine same, AGT code for serine that is also same, but here it is PCA coding for glycine. What happens? The GCC this will code for this, PCA code for arginine. So the amino acid changes, the expression itself changes. That is due to what? There is a shifting of a frame, a shifting of a sequence, DNA sequence shifting. Right? What happens? Only one base pair has been shifted here. Sometimes what happens? Again, the another pair is inserted, or sometimes the three different pairs are inserted. So this will add for some other protein. So that is a phenylalanine or any other amino acid. Take an example where the complete frame has been shifted. So even though it also causes less harm, but the whole sequence has been changed. So this is what we call as the frame shift type of mutation, where there is a shifting of frame because of insertion or repetition of the base pair that leads to the expression of a different amino acid. That is due to the translation of that particular RNA segment. Next one is the mutagens. We already studied these are the agents, the substances which cause mutation, right? So the name itself says mutation generating substance are called as mutagen. Agents, the substances that cause mutation are called as mutagens. One example is X-rays also cause mutation, UV rays, gamma rays, formaldehyde, even the mustard gas, all those cause different types of mutation. Among this, the UV rays cause a mutation called thymine dimer. So this A adenine always binds with thymine. So this is the normal feature. What happens in this case? Here it is a formation of thymine dimer. Here the T bonds with the thymine bonds with thymine itself. It is a mutation. It causes physical changes in the organism called as the thymine dimer formation. This is the mutation caused due to the mutagen that is of UV rays. There are other radiations which also cause different types of mutations in the organism. So the next one is of that is the frequency. The question. So we go through some of the questions. So here that is the first one. The first question regarding the concept that is the frequency of recombination between the gene pair. The frequency of recombination between the gene pair on a same chromosome as a measure of distance. So who measured the distance between the closely linked genes? He is the student of the Morgan, that is Alfred Stewart. What he says, the frequency of recombination between the gene pairs on a same chromosome as a measure of distance between the genes to map their position. It was used for the first time by scientist Alfred Stewart, who is a student of Morgan, who gives the concept of linkage mapping, also called as genetic mapping, called as linkage. Give the units that unit is called as map unit, also called as so the correct answer is it does a measure of distance first time by Alfred Stewart. Next one is a XO type of sex determination. X XO type of sex determination is according to the insects, particularly the jasper. So in case of Drosophila, it is of XX, XY type. In case of birds, it is of ZZ, ZY type. In case of monkeys also almost same, that is in case of fish also, that is male heterogametic. So the next question, there are two statements. So one is labeled as assertion, another one is labeled as reason. Right. So what it says is the Mendel's laws of independent assortment does not hold good for the genes that are located closely on the same chromosome. So the assertion is the Mendel's law of independent assortment that does not hold good for genes that are located closely on a same chromosome. Is it right or wrong? If the closely linked genes they are tend to separate or not, so their separation is their possibility of segregation is less because they are closely associated. So the statement is right. So the question assertion is right. The next statement says the reason says the closely located genes are independent. Is it true or false? Closely located genes are thought independent. Is it possible? If they are closely located, frequency of recombination is less. So that's why their possibility of assorting independently is less. So the reason is false. So the assertion is statement, that is assertion is correct, but the reason is not correct. So the answer is false. So the next question is again regarding the frequency of recombination between the genes. The recombination frequency between genes A and C. The recombination frequency between genes A and C. How much? That is of 5%. That means they are closely located. So that's why the frequency of recombination is less. So again, B and C, how much? B and C it is 15%. In case of B and D, in case of B and C, it is 9%. In case of A and B, it is 20%. Yes, it is 20%. And in case of C and D, it is A and B. So what you have to identify? What will be the sequence of these genes in a linear chromosome? What is the sequence of these genes in a linear chromosome? These are the sequence. What they are saying is sequence of these genes. How many genes are there? A is there, B is there, C is there, and D is there. What they are asking is the sequence of these genes on a linear chromosome. So the first one, to take in respect of any one of the genes. Suppose consider the gene A is located in this particular position, right? The gene A is located in this particular position. So what is the less frequency of recombination? That means more closely associated are having a less frequency, right? So only 5% that is of C, right? C is closely associated with the gene, right? Between A and C, the frequency of recombination is only 5%. That means they are more closely located, more linked, less recombinant. Next one, the frequency between B and C. First, we go through the B and D, that is only 9%. So A and C is 5%, B and C it is of 15%. That means B and C is of B, C is of 15. The next one is B and D is of 9%. That means they are located close to each other. So B and C is 15%. And A and B again C is 20%. In C and B it is 24%. Last one that is of 28%. Their frequency is more, their possibility to become the more. Frequency of frequency is more. So what is the correct sequence here then? A, C as well as B and D. So the first one is the correct based on their frequency of combination. So the next one is the experimental verification of chromosomal theory. So the answer for this is C.H. Morgan. So the theory was given by Sutton and Boveri, but experimental proof was given by that is C.H. Morgan. So the correct answer for this is Morgan, the third one. So the next question is chromosomal theory of inheritance was proposed by Walter Sutton and Theodore Boveri. Watson and Crick, double helix model. C.H. experimental proof of chromosomal theory. So Punnett, Dixon and related to the crossing Punnett square. Next one is of what map unit or also called as centimorgan is adopted in the construction of genetic maps. Here it is a unit distance between two genes representing 10% of crossover. Is it true? That is called as 1 centimorgan? No, because it is 10%. This is more a unit distance between the two extremes is 100%. Again, it is also not because it is not equal to 1 centimorgan. So the unit distance between the genes on a chromosome representing only 1% crossover that indicates 1 centimorgan. The correct answer is the third one. Again, here it is 50%. Correct answer. The third one for mapping. So the next question is about that is the frequency of recombination between gene pairs on the same chromosome as a measure of distance between the genes was explained by scientists. So what is frequency of recombination the gene pairs on the same chromosome as a measure of distance between the genes was first explained by Wayne Alfred Stewart to explain measure of distance in the form of map unit, linkage mapping. Explained by Alfred Stewart. The next one is of pedigree analysis. So what is that pedigree analysis? Here it is. It is knowing history of a inheritance of a family by drawing a family tree by using different symbols. So that is what we call as pedigree analysis. So first we will go through the symbols, then we will study the chart here. So there are some diseases. How they are transferred from one generation to another. So that is called as inheritance pattern. So that can be traced or tracked through the making a family tree called as pedigree chart, or the analysis of it is called as pedigree analysis. So what are the different symbols used here? The square symbol is used for indicating male individual. The circle symbol is used for indicating female individual. The affected individual females are indicated by the circle symbol. Right here, the square with filled with the filled square that indicates affected male. Right? So female, that is the circle. And the circle as well as square is connected, that indicates meeting between the two individuals. So if it is indicated with the two lines, that is called as consanguineous marriage. Consanguineous marriage or called as meeting between the relatives is called as is indicated by this symbol. So this is how the chart. This is first generation. This is the next generation. So this is the affected male. This is the affected female. This is the normal male. Normal male, normal female, affected male, meeting between the male and female, meeting between the relatives within the relationship. There is a process. Next one. So this diamond symbol indicates sex unspecified, and if those symbol indicating five, that means five unaffected individuals. Right. So these are the different symbols which are used to analyze the pedigree chart. Right. So the next part is about the pedigree charts. So if you see the pedigree chart, you can see that there are different types of disease. So there are different types of chromosomes. Autosomes are there, allosomes are there. So when it comes to the autosomes, the disease might be autosomal dominant, disease might be autosomal recessive, or the disease might be X-linked recessive. So when it comes to the autosomal dominant disease, that means if the individual should be affected, that should possess a dominant gene. That means if it is possessing a dominant gene, then it is disease. In please possess one dominant gene, then it is disease. If it is consisting of a recessive gene, then it is normal. So those type of diseases are called as autosomal disease, autosomal dominant. If the presence of a dominant allele causes diseases and the individual is affected, right? So next one is autosomal recessive. As the name itself is autosomal recessive. At least recessive gene must be only in a recessive condition. Only in a recessive condition. Is a disease. So if it is a dominant individual, it is normal. Again, if it is a heterozygous dominant, again it is a normal. So this is what we got as autosomal. When it comes to the X-linked recessive, again there must be a recessive. Suppose X carrying capital C, X carrying capital C, it is normal individual. X carrying capital C, X carrying small c, again it is a normal individual or also act as carrier. And the X is carrying C, X is carrying, then it is a right. So when these diseases appear, so we are going to analyze the chart. First, we will see the character. So when it is autosomal dominant, at least one dominant gene is present in the phenotype, right? So the disease appears every generation. So when you see the chart, the disease appears in each and every generation. Then it is definitely what dominant disorder. And the affected parents can have unaffected children. Parents may be affected, but the children might not be affected because of the presence of heterozygous. Right? Example, only one example is given in textbook, that is of myotonic dystrophy, example for autosomal dominant disorder. The next one is of autosomal recessive type. That means it only appears if the organism individual is having recessive genes for the character, that is of only in a recessive condition it will appear. Even if one dominant gene is present, that is normal. Right? So here it is. As it is a recessive, if it is a recessive, there is a skip of generation. In the parent generation, it may not be there. If it is in the parent generation, in the daughter generation, it will not be there. They skip the generation. The affected individual is not present in every generation. So it is a they skip the generation, and unaffected parents have affected children. Means they might come to the carrier. So unaffected parents can have the affected children due to the presence of allele. One example of autosomal recessive, that is of Kalsal anemia. Most of them are autosomal recessive given in the textbook. Kalsal anemia, Urea, then thalassemia, all these are examples of autosomal recessive disorder. When it comes to the X-linked, it will follow a crisscross inheritance, zigzag inheritance. That means it comes from female to male, then male to female as a carrier, then it follows male to male, male to male, male to male, that pattern. That is what we call this pattern. That is grandfather, and from the grandfather to grandson to the carrier daughter. It comes from grandfather to granddaughter through a daughter. That means from a male to female, then female to his son. That is, so it follows this pattern. Two examples, that is hemophilia and allergen. More males are affected because females are carriers here. The female affecting to a female is rare in this condition because they act as what? Act as a carrier. So we will study the pedigree chart. Here these are the autosome link. So when it comes to the pedigree chart, there are different types of diseases. Autosomal linked diseases, sex linked disease. When it comes to the autosomal linked diseases, there is a autosomal dominant disease, then autosomal recessive disease. So when it comes to the sex linked, again there is a X linked and Y linked. So when it comes to the X linked, again X linked dominant, Y linked. So the X linked recessive. So when it comes to the Y linked, it affects only males. If in a pedigree chart all the males are affected, only males are affected, then definitely it is a Y linked trait or a Y linked disorder. So we will see what are the genotypes possible for the autosomal dominant disorder to affect. So both are dominant, the person is. If at least one dominant, again the person is affected. If the genotype is of recessive, then the person is normal. So that is called as autosomal dominant. At least one dominant allele must be present in the genome. Autosomal recessive. So if it is a recessive condition, the person is affected. At least one dominant is normal. The two are dominant, then it is 100% normal. So these are the cases. When it comes to the sex link, so Y link, so that is only if the males are affected, definitely it is Y link. Females are not affected in this Y link. So when it comes to the X link diseases, again it is dominant. And as I already explained, if both are dominant genes, then it is disease. At least one is dominant, again it is disease. So again, if both are the recessive, definitely it is normal. If one is, in case of male, consisting of X chromosome consisting of small c, that is also a normal case. So again, in case of disease, dominant gene is present. These are the cases. Again, same case in case of recessive also. If the recessive gene is present, at least one recessive gene is present, definitely. If both are recessive, then only this disease. At least one dominant gene is present, definitely they are normal. Here, so there is a trick you have to know before the analysis of a pedigree chart. So if you remember this trick, you can solve any, how much difficult it is, you can solve this particular trick. So what is the trick? First you have to know all the affected individuals have affected parents. So first you have to look at the pedigree chart. Then you have to remember if all the affected individuals have a affected parent. So we will see, we will analyze through this one. So what is the first statement says? All affected individuals have affected parents. If the affected individuals have affected parents, what they say? All the affected individuals have affected parents. Here, affected individual, affected parent. Affected individual, affected parent. So again here it is affected individual, affected parent. So how many generations are here? This is the first generation. This is the second generation. Again, here it is. Both are the third generation. Both are the third generation because they come from the second generation. So those are the third generation. So what the statement says? All the affected individuals have affected parents. Affected individuals have affected parents. Affected individuals have affected parents. Yes, it obeys. Right. Again, we go through. All affected individuals have affected parents. If it is yes, definitely it is dominant. Either autosomal dominant or X-linked dominant. Right. Next one. Now we know that pedigree chart is dominant. Next, what you have to consider? The all affected male have affected mother. Whether we have to see that if all the affected male have affected mother, all the daughters of the affected male are also affected. So we will break down it. First, we have to see all affected male have affected mother. We have to see affected male have affected mother. See here, is the affected male have affected mother. Right. So here it is affected female, but these are affected males. Affected males do not have affected mother. All affected males do not have affected mother. Here it is one generation, affected male have affected mother. But here the males are not affected. Then the statement is not obeyed. Right? So the all affected male have affected mother. It does not obey. That's why it is a no. If it is no, definitely it will be a dominant, autosomal dominant. So by this method, you can analyze any of the pedigree chart possible. So you have to remember it. So we solve it through the normal method. See, as it is a autosomal dominant one. So if it is dominant, so there should be at least one, either it may be homozygous dominant or it may be heterozygous dominant. If it is normal, then it is definitely it is recessive. Here it is also recessive. Here it is recessive. Are not here affected, that may be the homozygous dominant. So what is the possibility here? Here we having this one or this one. So it must be having here it is BA. At least one dominant gene is having. Here it is normal. Here it is. Here we get a disease. Right? Disease female. So at least the male is recessive. So it might be heterozygous. Yes, yes, dominant. So that is dominant. So again, there is no skip of generation. The disease appears in every generation. Males and females are equally affected here. So it is also autosomal dominant disorder. So we will solve this one through that trick method here. First you have to see what? All affected individuals have affected parents. Affected individuals have affected parents. Here. Affected individuals have affected parents. Obeys or not? It will not obey because affected individuals are there, but they do not have a affected parent. They have a normal parent. Yes or no? It is no. It is no for a so affected individuals have affected parents. It will not obey. That's why it's no for the chat. So then definitely it might be recessive, right? The recessive one. Next, what we have to see? If it is recessive, we have to go through this. All affected female have affected father. Affected female have affected father. So affected female are there, but affected father is not. So here the male are affected. So all affected females do not have a affected father. If they do not have a affected father, it is yes or no? Definitely it is. If it is no, then it is autosomal recessive cat, autosomal recessive disorder. So autosomal recessive disorder, how we can identify with the normal method? So there is a skip of generation. Males and females are equally affected. There is no crisscross inheritance, so that's why it is definitely autosomal recessive disorder. It is of autosomal recessive disorder. As we studied, so this is of autosomal dominant. Right. Examples given for this is myotonic dystrophy. Myotonic dystrophy, related disorder. It is of autosomal dominant type. So autosomal recessive, most of this, such as sickle cell anemia, then thalassemia, as well as phenylketonuria, all those are what example for autosomal recessive. So in the NCERT textbook, these are the only types given. That is autosomal dominant, autosomal recessive, or you may get X-linked recessive, that is a crisscross pattern. So that is a hemophilia as well as color blindness. So we will see the types of diseases. There are two types of diseases mainly classified. One is the Mendelian disorder, another one is chromosomal disorder. So what are those Mendelian disorders? So Mendelian disorders are which follow the Mendelian inheritance. So that is of the hemophilia, it is X-linked recessive, sickle cell anemia, it is autosomal recessive, phenylketonuria again autosomal recessive, color blindness, it is also X-linked. All these are what examples of Mendelian disorder. When it comes to the chromosomal disorders that are caused due to the increase or decrease in the number of chromosomes, those are called as chromosomal aberration. Chromosomal aberration disorder. So example, Down syndrome, Klinefelter syndrome, another one is Turner syndrome. We want to study in detail. See, this is of different types of genetic types. One is called as Mendelian disorder. So here it is alteration or a mutation in a single gene. If any single gene are mutated, that causes Mendelian disorder. Chromosomal aberration, that is due to increase or decrease in the number of chromosomes, that leads to the abnormal arrangement of chromosomes. Those are called as chromosomal disorder. Among these, the first one is of autosomal disorders, another one is X-linked disorder. One is autosomal dominant, another one is autosomal recessive. So here it is autosomal dominant disorder. One example, that is of myotonic dystrophy. Most of the examples given are in the NCERT text, that is of autosomal recessive type. One is sickle cell anemia, another one is cystic fibrosis, another one is phenylketonuria, another one is thalassemia. Right. So when it comes to the X-linked dominant, so it is not mentioned, but you just know about it. That is vitamin D resistant rickets is an example for X-linked dominant disorder. When it comes to the X-linked recessive, there are only two given as an example. One is color blindness, another one is hemophilia. Those are examples for X-linked recessive disorder. As I said, chromosomal disorders, that means where there is a change in the number or a set of chromosomes, that is called a chromosome disorder. There are two types here also. One is aneuploidy, another one is called as polyploidy. So what is the meaning of aneuploidy? Whether there is a less number of one or two less chromosomes are there, or one or two more chromosomes are there. Aneuploidy is nothing but increase or decrease in the number of chromosomes that leads to a disorders. That disorders are called as aneuploidal disorder. So example for those is monosomy. So what is this monosomy? So chromosomes exist in a pairs. In any one pair, only one chromosome is present instead of two. So that leads to the 2n minus 1. One chromosome is less here. That's why it is called as monosomy. Another one is nullisomy. One pair of chromosome is less here. So that's why it is called as nullisomy. 2n minus 1. From a diploid set of chromosomes, one pair of chromosomes has been removed. One chromosome is less. So that is of nullism. Trisomy. Tri means 3. Instead of 2 chromosomes in a one pair, 3 chromosomes will appear. That is what we got as trisomy, indicated by 2n plus 1. Tetrasomy, 4 chromosomes will appear. That is of 2n plus 2. We are going to study chromosomal disorder. So when it comes to the polyploidy, whole set of chromosomes will become increased. In case of plants, it is observed more. You can see polyploidy there. You can see tetraploid plants. The wheat we consume, that is hexaploid wheat, has a 6 set of chromosomes. So that is what? A polyploidy, their whole set of chromosomes used. So that are commonly seen in case of plants. So this whole set of chromosomes. Next one is the Mendelian disorders. So when it comes to the Mendelian disorders, so first we will study the X-linked recessive disorders. The first one is of the color blindness. Color blindness, it is basically a X-linked recessive disorder. X-linked recessive, that means it follows criss-cross inheritance. It follows criss-cross. That comes from female to male. Then again, female becomes a carrier. Again, male. This. That is called as criss-cross inheritance. So why it is caused? It is caused due to the mutation in certain genes on X chromosome. It is caused due to the mutation in X chromosome. So that means there must be a presence of what? Recessive genes. What are the conditions here? If chromosome consists of recessive genes, in case of females, that is very rare. In case of males, if the X with small p and Y, then definitely it is a color blind. Both are deceived conditions. Even if it is presence of one capital C, one small c, it is a normal, but it becomes a carrier of the disease. It becomes carrier of the disease. Usually is normal. In case of female, the disease comes with rare here. There are two different types of color blind. Here, red color blindness is called as proteinopia. Red color blindness is called as proteinopia. And the green color blindness is called as deuteranopia. Deuteranopia. Right? It affects 8% of the male, only 0.4% of the female, because the females act as the carriers of the disease. It follows criss-cross inheritance. Right? As in males, only one X chromosome is there. That's why it is, if it is affected, males are colorblind. But in case of female, there are two X chromosomes. At least one dominant gene is present. The female will be normal. So that's why more number of males are affected than the more number of females. Right. So the next one, the same in case of hemophilia also. This is also a X-linked recessive disorder. So here what happens? The blood clotting factor is affected here. So the protein, it goes for a blood clotting that is affected. So if given a small cut, that leads to the continuous bleeding of the blood, cause to the death of a person. Here also more number of males are affected. So the small cut leads to unstoppable bleeding. It is also controlled by the alleles capital H, small h, where H is a recessive responsible for hemophilia, as it is a X-linked recessive disorder. What are the conditions here? P, X, H, Y, normal, X, H, Y, hemophilia, because small h indicates recessive gene. X, H, small h becomes a normal female, but it becomes a carrier of the disease. Here capital H is normal, capital H is small h, normal and carrier. H small h is diseased, that is very rare in condition. H is rare because the male should also, the father should also be diseased. So here, where it is appeared, it is called as a royal disease because it first appeared in Queen Victoria's family, where Queen Victoria was a carrier of this disease, and many of her descendants became hemophilic because of her, because he is carrying the diseases, having one normal dominant H, one recessive H that carries the disease. So female being hemophilic is rare, as he is affected father, where the male survival chances of male is very rare, because even a small cut that leads to unstoppable bleeding. Carrier mother to become a female. So it is very rare, because the father should be affected. So the survival chances are less. So that's why it is very rare condition. There is a so here. So this is a next one is of sickle cell anemia, that is of autosomal recessive disease. Sickle cell anemia. The name itself indicates here the RBC cells normal size, that is round and biconcave. So here they become a sickle shaped cell. So what are the genes that control that is where the person is affected? Both are both are carrying HBS, HBS are affected. Even HBA and HBS is a single pair of allele. It is a heterozygous condition. Right? HBA and HBA is normal. HBA as well as HBS is carrier. Both are affected. Both are recessive. Definitely it is a sickle cell. Here. So this is how sickle cell anemia takes place due to, as I said, it is caused due to what? Point mutation. So any change the unit that causes. Where on the sixth position of a protein chain, where instead of glutamic acid, the valine will be expressed. Here it is a GAG. Instead of GAG on the sixth position, what comes? GAG comes. GAG comes on the sixth position. Instead of glutamic, valine is there. So this valine causes a change in the state of the RNA. So this is the DNA sequence becomes RNA to translation. mRNA becomes a protein chain after translation. So then the GAG, instead of adenine, uracil will appear in the mRNA chain. So this converts into a valine instead of glutamic acid. So this one point of change in the nitrogen basis nucleotide is to another protein is expressed, that causes the shape of the RBC turn into a sickle. So it is caused due to what? That is also what? Autosomal recessive disorder. So there are other types of disorders. First one is phenylketonuria. As we already discussed earlier itself, is a autosomal recessive disorder, which is an born error of metabolism. So here, humans suffering from this disease lacks an enzyme called as phenylalanine hydroxylase. So why this enzyme is necessary? The enzyme is necessary to convert the phenylalanine amino acid into a tyrosine. If the person lacks this enzyme, phenylalanine is not converted into a tyrosine. Instead, the phenylalanine becomes a phenyl pyruvate that accumulates in the brain, causes mental retardation. And excess phenyl pyruvate that can be excreted through the urine. It also affects the skin coloration. So this is what one example given for pleiotropic disease also. Pleiotropy, single gene controls in character. What characters are controlled here? So here, mental retardation causes, these are affected, then the skin color is also affected. Next one is of thalassemia. So this thalassemia is also what? The autosomal recessive disorder. So it also can be caused due to mutation or deletion. See, there are two different types of chains are there in hemoglobin. So one is the alpha chain, another one is beta globin chain. One is alpha chain, beta chain. Based on that, there is alpha thalassemia which affects alpha globin chain, and there is a beta thalassemia which affects beta globin chain. See here. So in case of alpha thalassemia, which is controlled by two genes, one is HBA1 HBA2 gene here. So it is a pair of chromosomes that is affects on the 16 where HBA1, HBA2 are closely located on both the chromosomes. Each HBA1. Okay. So our diploid cells have two copies. That means both of the chromosomes have HBA1, HBA2, HBA2 protein on a chromosome number 16. So thus there are how many genes are there? So on one chromosome HBA1, HBA2. On another chromosome HBA1, HBA2. There are four HBA1, two HBA1, two HBA2 are there. That can cause alpha thalassemia. Out of these four genes, if more number of genes are mutated, if HBA1 is mutated, the person is less affected. If both are affected, then it is more affected. If the three of them are affected, more affected. So the more number of genes are mutated or deleted, more severe will be the thalassemia. Right? It affects the synthesis of hemoglobin that causes the abnormal hemoglobin. Quantity of hemoglobin is less if it is affected. That becomes thalassemia. So in case of beta thalassemia, which is controlled by HBB gene, that is hemoglobin beta globin cell, right? That is present on a chromosome number 11 here. Only one gene controls this beta thalassemia, HBB, located on chromosome number 11, right? So diploid cells have two copies of chromosome, so that's why two HBB genes are present. So if one is affected, the person is the condition is less severe. Both the genes are affected, the condition is more severe. It is quantitative hemoglobin disorder, right? So if more genes are affected, the severity is more. So that is also what? It is also an example for autosomal recessive type of disorder is caused due to what? The mutation or deletion of particular of these are Mendelian disorders. Now we will move to the chromosomal disorder. When it comes to these chromosomal disorders, first one there is a Down syndrome, and the next one is the Klinefelter syndrome. Another one is the Turner syndrome. First, the Down syndrome is also called as Mongolism, and where it affects the 21st chromosome. It affects autosomal trisomy. So the 21st chromosome is affected. So where instead of two chromosomes, two autosomes, one more is added. So the total chromosome is 46 plus 1, that is 47 chromosomes there instead of 46. So this was first described by the scientist Longden Down in the year 1866, where all the chromosomes were normal, but instead of two, one extra chromosome is added to the 21st pair, right? 21st pair, right? So one extra chromosome is added on the 21st chromosome, right? So it can occur in both male as well as female because autosome, it is an autosomal trisomy. It is same in case of both male and female. So what are the symptoms here? The affected child will have a short stature, stunted growth, height is less. Again, it has a small round head. The tongue is furrowed, more spirally turned. So that is having a P. And the mouth is always open here. In the next case, that is the continuous salivation. The saliva will continuously salivating out. So it has a broad palm with a characteristic deep creases are there inside the palm. Again, the physical and mental psychomotor is affected. Movement of the affected person is observed, so that it affects locomotion of the affected person and is mentally retarded. Again, it has congenital heart disease. So it is what? It is caused due to the appearance of one extra chromosome in the 21st pair of chromosomes. It becomes 3 chromosomes. So that's why it is called as a 21st trisomy. So it is autosomal trisomy. Next one is Klinefelter syndrome. So in case of Klinefelter syndrome, what happens? Normally there are 46 chromosomes, and among those 46, 44 are autosomes, one pair is allosome. But what happens here? One extra chromosome, female, so thus the males have overall masculine development, but there is a gynecomastia. That is, the development of breast occurs in males. As it occurs in males, what happens? So one extra X chromosome will appear instead of 46 XY. What happens? XXY will appear. One extra chromosome that causes overall masculine development along with gynecomastia. That means breast development takes place in case of males. So the males become sterile here. They are not able to reproduce. So again, we also have the mental retardation. These are called as Klinefelter syndrome, also called as allosomal trisomy. Also called as what? Allosomal trisomy. Here it is called as autosomal trisomy. Turner syndrome. Yes. So along with 44 chromosomes, there is one X chromosome is lesser here, and it is only seen in case of female. The female will have a rudimentary ovaries. Rudimentary ovaries, they will not produce egg cell. They are not able to produce egg cell, so they are just non-functional ovaries, and there is no secondary sexual character. So again, secondary sexual character will appear in female, and the females become sterile due to the rudimentary ovaries. This is called as the Turner syndrome, where 44 autosomes are there, but instead of two X chromosomes, only one X chromosome is there. That is the number 45. This is what the karyotype of the disease. Here it is called as 21st tribe. All these are what? Due to the chromosomal aberrations. Increase or decrease in the number of chromosomes. In case of Down syndrome, also called as autosomal trisomy, one extra chromosome appear on the 21st pair. Allosomal trisomy, on the allosome, one X chromosome becomes extra, so that is of allosomal trisomy. Example, Klinefelter syndrome. Next one is of Down syndrome, where one X chromosome is so these are the different types of diseases which we go through the different questions this time. So, which of the following occurs due to the presence of autosomal linked dominant trait? So, among this disease, you have to identify autosomal dominant disease. So, in the NCERT textbook, only one example given for autosomal dominant disorder, that is of myotonic dystrophy, and this thalassemia, this skull cell anemia, example for autosomal recessive disorder. And hemophilia and color blindness are example for X-linked, right? So myotonic dystrophy is the only answer. Here it is autosomal dominant trait. Next question is, if a color blind female marries a man whose mother was also color blind, so female is a color blind. So you already know color blindness is a X-linked recessive disorder. It is a X-linked recessive disorder. That means the female is color blind. That means she is carrying both the genes are recessive, right? So he marries a man whose mother was also color blind. So he will marry a man whose mother is also color blind. That means mother was color blind, marries a normal man. So the possibility is one possibility he is also what? He might also be deceased. So what are the chances of her pregnancy? having color blindness. So here it is the gamut. Here it is of them, all of the progenitors, all of the progenitors are color blind. Here is the possibility of all of our progenitors, 100% of them are what? 100% of those offsprings are color blind. So this is what it means here. The answer is 100%. Because the male is also affected. See here. Next one, in a cross between the male and female, both are heterozygous for sickle cell anemia. Both are heterozygous for sickle cell anemia. That is, both are heterozygous for sickle cell anemia. What percentage of pregnant will be disease? What percentage of pregnant you are asking? First one is it is normal because both are homozygous. Here it is E and S. Again, there is a dominant gene, heterozygous, it is also normal. So these are also normal. So only here it is the case. It is also heterozygous, they are normal. So the only case where it is a homozygous condition that causes the disease, that is only 25%. One of the four of our progenitor are affected. The answer to this question is yes, yes. That is only 25% are affected. So this much is all about the principles of inheritance and variation. It is also one of the high weightage chapter and one of the important chapter where uh, the questions almost you may get more than 4 to 5 questions from this chapter itself. So very important. Go through all these questions. Please you have to tell minimum 400 questions. This chapter, those are of different and a NEET level questions. Please solve those. Definitely will help for you. So it is one of the uh, high weightage chapter. So thank you for you all and all the best for your NEET exam.